surface area analysis. And so there is a difference. I like this cartoon. I think that my chemistry teacher actually used this, and I had to dig around to find it. But it does a good job where you have adsorption. So I also had a lot of people spell check me and say, you're misspelling absorption wrong. No, there is something called adsorption where it actually appears on the surface, and then absorption where it's actually entrained in the volume of the material. So if you have something that's absorbed, it actually goes into the volume of material, whereas adsorbed, you're talking about something that is on the surface of the material. And the way that it's explained in a lot of chemistry textbooks is there's what's called an energetically favorable condition between the molecules that are going to adhere to the surface of a particular material. So the process creates a film of adsorbate, the molecules or atoms being accumulated on the surface of the adsorbent. And so there's a lot of factors that go into this as to how well your absorbate will bind. And so it depends on the surface area, which we're going to talk about how you can derive or how you can figure that out. The activation, experimental conditions, especially the temperature and the pressure. And so if you have just a physical adsorption, that means it's not necessarily going to stay on the surface and be bound to the surface in a higher connection capacity, I guess you could say. But there's a lower heat of adsorption given off when the molecules actually combine or when they touch each other. So there's whenever the molecules do adhere, and they go through adsorption, there is some heat given off, and you can use some various types of equipment to determine the amount of, with the temperature increase, how much heat is actually given off at that stage. And with the chemisorption, where you have an actual chemical bond, and we talked about the difference between van der Waals and the chemical side of things, where you've got chemical bonds. So you're going to have a higher heated adsorption with the chemical forces going on, the chemical bonds. Usually the physisorption will take place at low temperature and decreases with increasing temperature, and the chemisorption will take place at a higher temperature, and with the chemisorption it's usually irreversible, whereas physisorption it's reversible. It's related to the ease of liquefaction. So what does that mean? Well, basically it's very pressure dependent. So if you have a higher pressure, you actually can turn your gas into more of a liquid state, and so it's very highly dependent on, the physisorption is very highly dependent on the pressure of what you're trying to measure, how well it adheres to the surface. Forms multimolecular layers with physisorption. You'll have molecules stacking, and there's a good picture of that here. So you'll have multi-layers that bind with physisorption, and it forms monomolecular layers, so it would just be like, you know, one of these balls here on the absorbent binding to that on monolayers. And then as far as energy it goes, with physisorption usually doesn't require much, if any, activation energy, and then with the chemisorption there is some that's required. So talking about adsorption models, I'm not going to ask you any crazy to memorize any of this stuff, any questions that might be out of the realm of, because this is more, I was trying to think some classes I've had that deal with this. Anyway, you can get into some really advanced equations to try to model how your adsorbent and adsorbate their relationship with each other. But we're mainly today going to be talking about Langmuir absorption model from 1918, and it's more of a monolayer equation that describes how well in this case, we're going to be looking at methylene blue. Methylene blue has traditionally been used to determine how well, let's say, your activated carbon will absorb various molecules. So to try to compare apples to apples throughout literature, they use different dyes, and different dyes are going to bind with different adsorbents course, differently. But to try to, instead of somebody using a diesel fuel from like the winter and somebody else using the same, and they say, oh, it's just diesel fuel to compare, diesel fuel mixture in the winter is not necessarily going to be the same as a diesel fuel mixture in the summer. So you can start getting into problems when you start saying, well, I'm comparing this oil, and somebody might be comparing the same kind of oil, but it's even a different season, and now you're getting completely different results than they did with your experiments with how well your material actually absorbs, let's say, diesel fuel or gasoline. Not all gasoline is going to be the same. Not all diesel fuel is going to be the same. Not all different chemicals you get, unless it's, you know, maybe from Fisher Scientific and 99.99% pure, you're going to have problems trying to compare your results with somebody that's done theirs in literature. So that's why traditionally there's, you know, 99.99% pure methylene blue dye is used, or Congo red. Different types of dyes like that are available so that you can do these kind of adsorption tests, and you can actually compare it back with literature to see how well your adsorbent works compared to what somebody else has done in the past. Does that make sense? Even when you get into, like, some people say, well, we use a transmission fluid, or we use a differential fluid with this particular company. Well, even that company, they may have changes in the amount of whatever they're using. Maybe it's a surfactant or something to keep the material, try to keep the oil clean. So they may have changes in their chemistry of their product. So that's why this is done this way. All right. So we're going to be talking about this one. Next class we'll be talking about using BET theory. But today it'll be a little simpler. I say simpler. It's good. I'm going to record this and upload this because this is a lot of information, especially trying to determine how well an absorbent works. And the reason this is important is because as you're producing different side products, let's say you're at Weyerhaeuser, for example, with their continual dry kilns. They got continuous dry kilns that produce, they're using a gasifier to heat their continual dry kilns. They have a lot of biochar that comes off of that. And that biochar can then be activated to produce a very expensive activated carbon, relatively speaking to biochar, and activated carbon is very expensive. So that activated carbon could then be sold to water filtration companies. I mean, I'm buying a water filter for my refrigerator for the water you get out of the fridge, and I think I'm spending about $40 every 5 months or something, 4 months. So, you know, it's used, and that's a small way it's used. It's also used, and activated carbon is used for major water filtration companies when they're doing water treatments. It's used in the alcohol industry if you're trying to produce specific alcohols and you're trying to dehydrate the alcohols that have a lot of water in them, and you're trying to get as pure as you possibly can alcohol content, and just trying to remove water. And also trying to filter out contaminants. So the Department of Chemistry is doing work where they're taking biochar, activating it with higher temperature, and producing this activated carbon that can then adsorb lots of different heavy metals, and phosphorus, potassium, lots of fertilizers that can then be recycled back onto their farmland. So there's a lot of use, a lot of good applications for this, but trying to figure out how well your particular thing or your absorbent that you're making measures up to what's already traditionally out there is important to just make sure it's economically feasible and that you can compete with other folks that are making this stuff. So methylene blue. I think y'all may have already seen a video, but I like this. This is a really short video that describes how a spectrophotometer works, and it's extremely important in this field for determining your concentrations. Let me go back.
Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism, and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
All right, so let me go focus in on this test tube here.
Light being transmitted through the sample.
All right, so in what the experiment I'm going to talk about, what they've done is they've put various concentrations of methylene blue in a larger container, and then they've dropped in activated carbon. Then after mixing it for a certain period of time, they can then take so imagine they've got the big flask or beaker here. So then they filter out the activated carbon that remains, and they take a sample of that liquid that's in their bigger container. It's usually about 10 milliliters. They'll take that sample and put it into this, what they call a cuvette, which contains about 10 mils. And that cuvette is what you're measuring. You're measuring to say, okay, I've put in a certain amount of activated carbon in this big container to adsorb the methylene blue that's in there. And I know and you measure out and you know there's a specific concentration of methylene blue, whether it's 100 milligrams per liter or 500 milligrams per liter, you do that. So you've got a certain amount of concentration of methylene blue in your container. Then you throw in this case, activated carbon, and that activated carbon is going to suck up some of that methylene blue. That adsorption effect is going to take place. Okay? So the adsorption of the methylene blue on the activated carbon occurs, and so now when you take a sample of that liquid, are you going to have less or more concentration of methylene blue in your cuvette? You're going to have a greater concentration or a lower concentration if your activated carbon picked up some of the methylene blue? Lower, that's correct. Hopefully if everything went according to plan and you don't have a lot of error, you have a lot, and that's another thing with this you have to be careful about with these cuvettes. Usually they actually sell some that are disposable because you can have some residues if you're not very careful. And actually I have seen some experiments where they measured more and I said, this doesn't make any sense. How do you have more concentration? That goes beyond all logic, right? So when you have those situations, you have to be careful of error because you can, in the pipettes that you're taking, if you reuse pipettes or if you reuse the same cuvettes that you're using for your experiments, there can be a lot of error there. That's why they do sell quartz ones where you have to be very careful and wash them out really well, but they also sell disposable, they call it disposable cuvettes, so that kind of error doesn't happen. But yes, you should have less of a concentration if your activated carbon has sucked up the methylene blue like it was supposed to. All right, so that's what they're doing. They're measuring in this, and I think I've got the YouTube video somewhere. It's maybe in the notes, but you can go through and watch the full thing. Here, let me see if I've got it in the notes. It's in one of these. Yeah, well, it's below. It's on this slide number 16. They go through the YouTube video and explain exactly what they're doing, but I'm just, for sake of time, I'm just going to try to summarize what they're doing. So in this experiment, they're going to be using a spectrophotometer to determine the concentrations, the constant stirring equipment, and then a weighing balance. So they have a certain amount of distilled water they're using in a beaker, and then different concentrations of methylene blue that they weigh and then put into these beakers so that they have a specific concentration of methylene blue in milligrams per liter. And they're stirring their samples at 150 RPMs until equilibrium is attained. I'm not sure how many hours they use. I have to go through the video again. It's been a while since I've watched it. But in many cases, equilibrium, many papers quote equilibrium time as being at least 48 hours. I think they may have used less time here, but if you're going to try to get a paper published and you're talking about the equilibrium time that was used for your activated carbon or whatever your adsorbent is, you would usually let it go for 2 days to just be sure that the activated carbon has sucked up as much of the methylene blue as possible to reach what's called equilibrium time, or your equilibrium of your material. So the samples were collected and measured at a wavelength of 665 nanometers. So that's that wavelength that we saw in that video. There's a specific wavelength that's needed for methylene blue, and that's 665. If you look it up on Google, that wavelength corresponds to a blue color. So makes sense, right? All right, so you're looking for blue. All right, so and then you obtain calibration values with the known concentrations that you're adding Similar to the way we did the calibration curve for the gas chromatograph, they're doing a calibration curve for their spectrophotometer. So this is what their procedure was. They added 0.2 grams of activated carbon. They agitated the sample until their equilibrium is achieved. They filtered it, collected. They just used within 10 minutes. And then this was measured using a spectrophotometer. And then, actually, this is minutes at the bottom, if I remember right. And then the removal efficiency. So it looks like after 15 minutes, they basically have for everything that they've got there, they're removed. That means they removed about 95% of the methylene blue that was contained in that beaker. So it worked really well after about 15 minutes. Then they plot concentration of methylene blue in the solution versus time. You can see it kind of changes depending on how much material is there. And then they had their concentration over their initial concentration versus time. And there was a little peak there with the 10 milligrams methylene blue, and they said that it's either experimental error or there may have been some desorption that occurred, and then it resorbed on the surface. Usually you would have peak or lines that look like the red and gold or green line there, ideally. But there may be some desorption that occurs with the smaller amounts of methylene blue just because there wasn't a whole lot available there. Yeah, it's hard to say exactly why that occurred. All right, and then here they're plotting the mass of methylene blue absorbed per mass of activated carbon that they used. So in this graph, for the blue, after, 30 minutes in the blue, they had about 100 milligrams of methylene blue that was absorbed on 1 gram of activated carbon. You see how they plotted that? Okay. And then with the red, about 30 minutes, they had about 290, and then with the green, 50 milligrams of methylene blue initial concentration, they had about 450. All right. So these are some equations that you need to know for these next graphs that they've got here. So C naught is the initial equilibrium or initial concentration. So that's this initial. So that's the first thing you're using or you're putting into your beaker. Equilibrium, in this case, is where they're saying equilibrium is I think 60 minutes or something, but ideally 48 hours. If you're doing this to try to publish in a journal article, you want to try to leave this mixing in your beaker for 48 hours. So that's the concentration that you would measure. After 48 hours, you put in your spectrophotometer with your calibration curve, and you would have your concentration that would come off of your spectrophotometer. And then at a specific time, they have CT. So they're going to be plotting a lot with QT and QE. So these were the equations come from. So in V is the volume of solution they used, and W is the mass of the dry adsorbent that they used. So that's where these equations came from. And we'll look at what these mean on these graphs, but if you're plotting it as what's called a pseudo first order, it's that physiochemical, the van der Waals interactions. Or if it's pseudo second order, then you have chemisorption. You have those chemical linkages going on. So if you plot this with log QE minus QT versus time, you end up with this kind of graph where there's not really, if you try to plot an R squared of this, you'd have some problems. You wouldn't have a very good coefficient of determination. It's hard to say what's going. Basically, this is telling you this is probably not a physiochemical interaction. You're probably not getting the van der Waals interactions. But if you plot T divided by QT versus time, that's what's called a pseudo second order And those look better. So therefore, because if you're plotting this T over QT versus time, because it's the second order, it's a chemisorption interaction. You can determine based on these graphs that you have got, let's go back to the beginning. You've got this type of interaction going here on the right side versus the left side. And that's just solely based on these graphs where you're plotting. You're wondering where did this come from on the y-axis, and why are you plotting at this versus this? Well, there's equations that are set up for that, and that is this Langmuir equation here. That's where this is coming from. This adsorption model here. That's why you're plotting it that way. And then we'll go through a journal article, and so I just wanted y'all to see these first before we go to that journal article. That based on these graphs, the log QT minus QE minus QT on the y-axis, and then the pseudo-second order where it's just you're plotting T divided by QT. Based on just those different y-axes, you can determine whether or not you've got a first order based on if your R squared value looks like it's 0.9 something, 0.92 is probably pretty good, versus this is probably more like 0.1, if that, for the green. Well, green would probably be more like 0, and blue, I don't know, blue's maybe 0.1 or something, but you'd have to, you can right-click and draw a trend line, but it's not very good. Basically, that means 10% of the time your X and Y axis are going to be, you'll be able to draw a relationship. You could flip a coin and get better results than that, right? So that's not a very good way of modeling what you've got going on, whereas this is. I mean, the plots and the lines line up really well. So just based on what your y-axis are, you can determine if it's a physiochemical or if it's got a chemisorption pattern. Everybody okay with that? Anybody have any questions so far? I'm going to try to cover this a little bit more in depth when we go to the journal article. Okay. So that's what this is saying. If your coefficient determination is really close to 1, and you've got that pseudo first order reaction with that y-axis of the log QE minus QT, then you've got physisorption, and then the second order you've got chemisorption. All right, so that's great and all. You can tell which one you've got, and if you've got a strong interaction. Most of the time with methylene blue, with activated carbon, you're going to have that chemical bond interaction of the chemisorption. But if we've got the question really becomes, how much methylene blue can I get to stick to my activated carbon? And that's really what it boils down to. And that is probably the most important question whenever it comes to trying to compare apples to apples. How do we determine how well your adsorbent is really working? And so that's what I'm going to show you on this Excel file that we're about to look at in this journal article. So in this AMID journal article, in this group, they studied bamboo activated carbon. So let's look at that article real quick. All right, so how did they do this? Well, they activated basically they activated biochar, bamboo biochar, which you just heat. I don't know what their condition for the biochar was, probably about 400 to 450 degrees C in the absence of oxygen. So they made this biochar, and then they used potassium hydroxide and carbon dioxide as activating agents at 850 degrees C for 2 hours. So what you're doing there with a biochar, you don't have a whole lot of pores. You don't have a whole lot of surface area with just biochar. You can activate it by throwing it into a very basic solution and then also heating it at really high temperature. I actually got a paper where we studied doing it like potassium hydroxide, then we did a really strong acid, and then we did just heat with, I think it was just pine biochar. But there's a lot of different methods you can use to try to basically, you're trying to make holes or pores form in that biochar to make it a very expensive adsorbent. And then these adsorbents can then be used for oil spill cleanups and water filtration and all that stuff we talked about. Or trying to collect fertilizer that runs off of farms. So there's a lot of good uses for this. And then, so what did they do? So we're going to look at, they did a time study similar to that PowerPoint I showed you, but how do we get to this number that they got here in their abstract? They said we've got an adsorption capacity of 454.2 milligrams of methylene blue can be absorbed on 1 gram of activated bamboo char. So how did they get to that number using methylene blue? Well, that's what we're going to look at today, how they got to that number. So if you're ever wanting to know how well you've got your adsorbent works, we can do that. So first of all, to get that number, you really need to look at the equilibrium study. And so they talk about they used 43 Erlenmeyer flasks, 250 milliliters, were solutions of dye with a different initial concentration. So they used 100 to 500, let me zoom in on that, milligrams of methylene blue per liter were placed in these flasks. An equal mass of 0.2 grams with a and that's important too, very specific particle size. If you use various particle sizes all over the place, you might get all kinds of crazy different answers. So they used their activated carbon was sieved at a very specific particle size, 150 microns, was added to the dye solutions and kept in an isothermal shaker at this particular temperature for 48 hours to reach equilibrium. And then they also used a blank for these solutions to make sure everything was properly correlated or what's the word I'm looking for? Well, where they can compare the blank to what was going on there. And the calibration, there we go, calibration. Their calibration. All right, and then the pH was adjusted to 7. The flasks were then removed, and then the samples were filtered, and they have each experiment was duplicated under identical conditions. And so then they show this is the main equation that we're going to be using in our Excel file, where the CO and CE are the concentrations of dye at initial and equilibrium. And then the V is the volume, W is the mass. All right, so let's look at the Excel file and walk through what they did. And they didn't actually publish the exact, the hard numbers. So I did my best to look on their graph and try to use a ruler and get the numbers that they had for their results. So these results might be a little different than what are in the article, but I'm just trying to use this as an example to show you all what they did. So there might be a little bit of discrepancy on the exact numbers, but the procedure is still the same because they didn't publish the exact numbers in a table. So I had to kind of look at it on their chart that they had. All right. So here's what we're dealing with. We got QE, which is this in this equation, our initial concentration, equilibrium concentration, the volume, and then the mass of the adsorbent. So here's what they had on their table or their chart that they had. They had these initial concentrations. So they're putting 100 milligrams of methylene blue per liter. These are their concentrations that they use, 200 milligrams per liter, all the way down to 500 milligrams of methylene blue per liter. And then they let it sit and mix with their 0.2 grams of activated carbon here, 0.2 grams of activated carbon in that solution for 48 hours. Okay, then they measured and found these numbers. So these are this C naught and CE naught, you know, because that's what you measured. This is what you picked out with the spectrophotometer after 48 hours. You got these. So after 48 hours, you didn't see anything. That means what? If you've got 0 concentration, what happened? What? It absorbed it all. That's right. Your activated carbon did its job, absorbed it all. And then at 200, not as good, but still pretty good. I mean, that's pretty good. So and then you get to 500, it's 59. So then you just take these numbers and you plug it into this equation. So all I'm doing for QE is I'm saying C naught minus CE times the volume, then divide that by the weight or the mass. All right, and then I get these numbers. So I do that all the way down. Does everybody see where the QE came from? Not too hard, right? Okay. Then I can do a plot of CE. So this is just a plot of your equilibrium concentration plotted to your QE. And I just wanted to make sure that my graph looked like their graph. But what we really need to do to actually determine the amount of absorption capacity is we need CE over QE And that comes, the reason why we're plotting that is it comes from this equation here, the Langmuir equation. So that's why we're doing this, is we're basically taking this equation and we're trying to get a slope, an inverse slope of, if you notice, this is like a y equals mx plus b kind of equation. And so we're trying to find this number as our slope, which to then ultimately derive Q naught. Where that's what we're looking for, is this Q naught, and that Q naught will tell us what our milligrams of methylene blue can be absorbed on our 1 gram of activated carbon. All right, so as we plot this, we end up with this graph over here. I'll just do it again here. So if we select these numbers and we go to, hey, look at this computer, it'll probably be better. If we go to insert, just a scatter is fine. Well, we'll do a line. Line, and we right click and we hit add trend line. We want our equation and we want an R squared value. That's a really good R squared value. So now we have that slope we were looking at. But that's not the real, that's not Q naught. Q naught is 1 over that number. So we can type that in here. 1 divided by 0.0022. There we go. That's our maximum monolayer adsorption capacity based on this Langmuir equation. And now we have our Q naught value. And that's really how you can mainly compare what your how well your material, your absorbate, compares to somebody else's. Does that make sense? I just kind of ran through that a little quickly, but you're using this equation to find the slope. And so when you plug in CE, the equilibrium concentration, versus the equilibrium concentration divided by QE, you get this equation, which is neat because then you can just use the inverse of that slope and get your monolayer adsorption capacity. So as we look at that, go back and we look at this article, they list theirs versus a lot of other people's in different references. So basically what they're saying is the way that they treated their bamboo was really good, except when I guess when you compare it to straw. They had 472, they had 454. But it beats a lot of these that were the methylene blue. You can't really, I mean, you can compare the basic red and these other ones, but it's better to compare apples to apples with methylene blue. So you really, to really try to compare the work, you just really need to follow the methylene blue listings here, not the basic red stuff. So you can compare one dye to the other. And it looks like it beats out just about everyone except for that one there. Everybody understand what they were doing here? Anybody have any questions? Yes. And I like doing it this way because you can also, we're going to talk about BET next lecture where we're looking at the gas adsorption instead of the liquid absorption. But if you're just trying to quickly determine if what you've got would compare well with the liquid absorption with something else, this is a really good and cheap way to do it as compared to the BET. I think Dr. Hassan told me just to run one sample of BET is like $150 or something. Whereas this, you can buy like a 10 kilo, well, maybe not that many much, but maybe 5 kilos of methylene blue for, I don't know, $100 or something, or maybe not even that much. And then you saw how much they used. It's not very much. You're talking about milligrams per liter. That 5 kilos would probably last you for 30 years if you were, just doing it off and on. So it's a good way, in my opinion, to do an initial check. Or if you're just concerned about how well your absorbate will work to absorb liquids, then it's a good method to use, and it's very publishable. This data is very publishable to try to determine how well yours works versus somebody else's. The other thing that they did is they did that time study where they did pseudo first order kinetics. So you can see there they had a pseudo first order kinetics just similar to the way that we saw on the PowerPoint, and then they had their second order kinetics. I think they drew the line here. Yeah, so their Suter second order kinetics, wish they would have put their R-squared values up on the charts, but it looks really good. And that's what you expect with methylene blue and activated carbon. You expect the chemisorption to go on. Yeah, that's first order. Not so good. Not as good, for sure. Had something there and there and there. All right. Any questions? And their graph up here also followed very similar to what they had in that PowerPoint experiment. They had time in hours here, where you had a higher, as your concentration went up, it makes sense that you'd have a higher milligram per gram absorption on your activated carbon.